His slides are working. Start that there.
And Richard doesn't need introduction to people here because he's one of our own. Richard received his MD from NYU in New York. He's a New Yorker.
Change the tab.
Received it from NYU and then came to Washington University, did his residency here and fellowship here with in cardiology with Bert Sobel, right? Yeah, Bert Sobel. And I think about 6 years after finishing his MD, he also did his Ph.D. here in chemistry. So he's on both campuses, as you can see, and he really integrates medicine with basic science in biochemistry and chemistry, and he'll tell you exactly what he does, because he knows it better than I do.
Well, thank you very much for inviting me here. So, by way of disclosure, this was language that was made by our Conflict of Interest Committee. So I'm a consultant and have an equity position in lipospectrum, and I'm president of Platomics, and I hope I'm doing a better job than the current president that we all have to serve under. Today I'm going to talk to you about biologic membranes, and there are three facets of biologic membranes that I'd like to stress. Biologic membranes are composed of, as you all know, a membrane bilayer, and they have transmembrane proteins in them. They're scaffolding for self-organizing chemical assemblies. For example, many ion channels are self-organizing chemical assemblies. Their scaffolding and their purpose lies in the fact that they undergo rapid lateral diffusion on the order of 10 to the minus 6 centimeters per second, so that everything now is in a plane. Two molecules that want to become partners diffuse in the same plane, and it's a two-dimensional diffusion problem, not a three-dimensional diffusion problem for a reaction to occur, or for two partners to meet and stay together. They're a bilayer medium for membrane trafficking. The Nobel Prize in Medicine was given 2 years ago for membrane fusion, and basically all membranes start out as basically phospholipids that get organized into bilayers and then get processed through the secretory pathway. And third, and what I'm going to focus on, is that they're a storage depot for latent cellular chemical messengers. So these guys here, the little sticks coming down, okay, those are aliphatic chains. Many of those aliphatic chains in biologically important membranes are comprised of polyunsaturated fatty acids that are then the targets of oxidases, which turn them into specific either eicosanoids or docosanoids, which interact with over 200 eicosanoid receptors. So in the beginning, okay, the field seemed to be very simple. That the arachidonic acid in cell membranes was hydrolyzed by an enzyme known as phospholipase A2 into arachidonic acid and a lysolipid. Okay, and this is a chemical reaction that is very straightforward, right? It leads to the production of two metabolites, including the liberation of arachidonic acid, which is now susceptible to oxidation. Okay, it was not accessible to oxidative reactions when it was esterified to the carbon and glycerol backbone. And then these eicosanoids, this arachidonic acid, can then get processed by dozens of enzymes, only a few of which are shown here, to generate signals, okay, generally through receptor-mediated events. So all was well and good, and that was the Garden of Eden. Everything made sense and interdigitated perfectly. Now the question was, did you want to take a bite out of the apple and learn more? Because it was a dangerous situation. I was brought up thinking or being taught that the phospholipases that generated these lipid second messengers were the same phospholipases that were present in snake venom. And snakes, as you know, will bite you and inject their venom extracellularly. And I could not imagine that the same type of enzyme was present in mammalian cells, which needed to be precisely regulated in a spatio-temporally organized fashion. So I took a bite out of the apple and was still wondering whether that was a smart or healthy thing to do. So I tried for a long time to get heart cells, fibroblasts, and other molecules that were presumably the mediators of many physiologic and pathophysiologic processes, but I had absolutely no success. Chandra remembers a time when I worked for 2 years and I didn't have a successful experiment. These days you'd have thesis committees, you know, like running all over you in the same context. So I decided to start from the beginning and actually find out what was present in heart cells and what the substrates for heart cell phospholipases were. Not knowing what phospholipases were present, but I was successful in being able to identify the first lipidomic. I was successful in being able to analyze through lipidomic technologies the very first mass spectra of ethanolamine glycerophospholipids and choline glycerophospholipids. So now, all of a sudden, we knew what molecules we were dealing with. Everybody had taught us that there were diacyl phospholipids. And if you look here, which is on the left-hand panel, which is now colorized like Gone with the Wind, because this was done in 1984, okay? These three peaks in yellow, okay, correspond to plasmalogens. They have a mass defect of 16, okay, because they're missing an oxygen atom with the same number of units of unsaturation. Moreover, okay, at R2, plasmalogens contained almost exclusively arachidonic acid. Okay, so they were the main reservoir of arachidonic acid that people were claiming to be liberated from the diacyl species. So this was somewhat of a revelation to us. We had been using the wrong substrates. We needed to develop synthetic methods to generate plasmalogens. And we now had a direction and an understanding, at least in part, of why our experiments hadn't worked for 2 years at all. So we worked for 5 years purifying the phospholipases from heart in a project that I wouldn't even dare to recommend to a graduate student. It took 5 people working probably 14 to 20-hour days, 5 years, to purify the first of this protein, HPLC beta. This protein is remarkable for the presence of ankyrin repeats, an ATP domain, a serine lipase domain, and a calmodulin binding domain. We cloned this protein from the sequence that we got from our purification methods. And this protein now is known to be the cause of infantile neuronal dystrophy, axonal dystrophy, Parkinson's disease, and it's also been implicated in Alzheimer's disease. But these top two are, the top one especially now, there have been hundreds to thousands of cases of INAD, which are due to HPLC beta. Parkinson's disease has a very strong GWAS association and was implicated by Vittorio Valentino in Parkinson's disease and capacitative calcium influx, which we'll talk about in a few minutes, more than a few minutes, 10 minutes. Now, once we had the structure here, then this got fun. So we put in 5 years of hard labor in Siberia in the Gulag, right? And we ended up with the structure. And this was in the days of the Genome Project, right? So the Human Genome Project had data streaming online, right? So all we had to do was wait until some data came showing that the ATP binding site and the lipase site were approximately 20 amino acids apart, and then we knew we had a really good chance of finding another phospholipase just by cloning that gene out. And in fact, this became very successful. We found IPLA2 gamma, which is responsible for ataxic neuronal dysfunction, generates mitokines which act, which are generated in heart mitochondria and act distally, or in distal membrane compartments or other cells. And then there are the 3 triglyceride lipases, which are also present in this family. The first one has a number of pseudonyms. We call it IPLA2 epsilon, but other people, right, and the majority of people will call it a adiponeutron. A adiponeutron was found by Helen Hobbs to be responsible for the release of fatty acid from triglycerides and fat droplets. ATGL was also present in this initial screen. ATGL stands for adipocyte triglyceride lipase, and this is responsible for the hormone-sensitive or isoproterenol-induced release of fatty acids from triglycerides. And IPLA2, which is also known as GS2, and its function is unknown. Then the rest of these enzymes generating the PNPLA family, patatin-like phospholipase family, have some diseases associated with it, and these 2 remain unknown. So that was, I think the principle of this was that if you work hard and identify something new, and then you exploit that knowledge in the appropriate way, you can get a lot of mileage. Clearly, science is nonlinear, and this is one of the best cases of nonlinear science that I'm aware of, at least in my experience. So if you look at the major phospholipases on the 23 chromosomes, you'll find that CPLA2 alpha, there are 2 families, the cytosol, the calcium-dependent phospholipases, and the calcium-independent phospholipases. Calcium-dependent ones are noted by a C, and the independent ones are noted by an I. What I showed you before, all related to the I's, okay, calcium-independent phospholipases, but in fact, this always happens. But there's another. I think I took that out because this intermittently loses its energy. Can you explain that physically? Okay, so the CPLA2 enzymes, okay, are the calcium-dependent phospholipases. And the calcium-dependent phospholipases, or CPLA2 alpha, which everybody thinks is the canonical phospholipase that's responsible for everything, but it is not. It's responsible for a large number of important processes. But the other enzymes in this family, CPLA2 gamma, thank you very much, CPLA2 gamma and CPLA2 zeta are very important and will be discussed during this talk. Two-handed. Okay, so let's talk about the enzyme that took 5 years to clone and the chemical biology. So the first thing that we thought was very interesting is that this is an initial rate, okay, of enzyme activity. That is the release of fatty acid from the SN2 position. And you can see that the release is linear over a minute, okay, and it's increased about 4-fold using ATP or ATP gamma S. So it's regulated probably through an allosteric mechanism by ATP, right? And I know there are a lot of people in the electrophysiologic sector, right, that think ATP is a very important modulator, and I believe that's probably true in membrane chemistry as well, and it may not be a coincidence. The next thing that was very remarkable about this enzyme was that it was activated by calcium and in the presence of calcium and calmodulin. Calmodulin became activated and ended up clogging the active site. Okay, so this enzyme is inhibited by calmodulin, by calcium-activated calmodulin. But if you incubate it in the presence of acyl-CoA, okay, which also has 3 negative charges, a ribose ring, right, and has a structure not so dissimilar, right, from ATP, this inhibition goes away and the enzyme becomes activated again. So now we have a war, right, between the calcium-activated calmodulin and the acyl-CoAs amongst other post-translational modifications, which determined activity. So this is a slide that I'm sure is familiar to most of you in the audience. And this was an important slide, actually for me, because it showed, I think, that membranes and ion channels are intimately related, and that ion channels do not exist in a vacuum or the gas phase, but are part of a membrane, and the biologic characteristics and physiologic characteristics and parameters of the membrane can indeed influence gating currents, channel function, and this was a hard concept to get by and to convince electrophysiologists of in the 1980s. When Roderick McKinnon came along in the mid-90s and started crystallizing the potassium channel, and finally in his seminal papers in 2000, I believe this was 2002 that this paper came out, the existence of channels causing alterations in membrane structure or membranes causing alterations in channel function were, I think, definitively proven. We had taken some proteins and renatured them in different phospholipids, and we showed that they had different electrophysiologic characteristics depending upon whether there were plasmalogens there, that was the molecule without the oxygen, or diacyl phospholipids. So the surrounding phospholipids in an ion channel have a great effect on the conductance and the properties of the channel. Now, in 1995, when we discovered that acyl-CoA could prevent the calmodulin inhibition, we came up with this model for store-operated calcium entry. And for those of you who are unfamiliar, what is store-operated calcium entry? So store-operated calcium entry is when the endoplasmic reticulum, the calcium stores in the ER or SR, go down by IP3 stimulation. Calcium's released, the calcium goes down. And what we had proposed was the calcium going down in the SR or the ER, resulted in, the decreased activation of calmodulin, leading to IPLA2 beta. And we said that lipid second messengers were generated. This was in 1995. We really didn't know what lipid second messengers were generated, who were the actors, nor did we know the ion channels that they were acting upon. But as the years passed, things became more and more clear until we now, I think, have a pretty good, although not definitive, molecular understanding of the importance of capacitive calcium entry. There are other models which basically, I think, state the same thing, although the spatial temporal location of these processes. This is now in a paper by Victoria Volatina, who I mentioned before, in 2012. And Victoria liked to think of the fact that IPLA2 is at the plasma membrane producing lysolipids that then activated ORI1 and calcium and sodium entry. And she recently published a paper showing that the defect in Parkinson's disease, in a model of Parkinson's disease, was defective calcium store entry. So Victoria's model basically, has the IPLA2 at the plasma membrane, there's IPLA2 at the plasma membrane, and the actors being lysophospholipase here. And finally, there's the arachidonic acid activated pathway of Trevor Shuttleworth, who was able to add arachidonic acid, and the arachidonic acid then stimulated what his channel was, which was a combination of ORI1 and ORI3. So it's not just ORI1 like it is in calcium, but it's not just ORI1 like it is in the Volatina model, it's a combination of ORI1 and ORI3. And Trevor Shuttleworth also was the first one to recognize the fact, or proclaim the fact, that STIM1 was not only present in the subsarcolemmal calcium stores, but was also present, about 25% of it was present in the plasma membrane where it was allowed to interact with ORI1 and ORI3 to generate a calcium current. So although the exact details aren't known at the present time, these three models obviously have a lot to do with each other, and I think the definitive anatomical relationships of each of these two others will be established. I don't see any reason a priori why all three can't have, be effective in different pathophysiologic situations. But I'm sure this will be the source of controversy for years to come. So, what about calcium-independent phospholipase A2 can be interesting. So we used here a gain-of-function model where we specifically transfected cardiac myocytes with IPLA2 beta. So transgenic models tell you what the enzyme is capable of doing. They don't tell you what the enzyme actually does. For that you need to do a knockout, and even then you have to be careful with your interpretations and provocative maneuvers. So using the gain-of-function model, we basically just did the standard gain-of-function, putting it in front of a MHC promoter, okay, and screened the newborns, and got a mouse that made about 5 to 10 times more phospholipase A2 beta than the wild-type mouse. And here's the interesting part. Okay, so this mouse, the wild-type mouse, right, control and transgenic, okay, there's not much difference. If anything, the transgenic mouse may have a little bit lower arrhythmias, just, at the time of ischemia. But now as you go out to 5, 10, and 15 minutes, which are the most likely times for you to develop VT, which is very difficult to induce in a mouse, as many of you in the room can attest to. There are multiple episodes of PVCs, ventricular tachycardia, in the basically the 5 to 20 minute range. Now the remarkable thing here is that we made an inhibitor, a mechanism-based inhibitor called BEL, which stands for bromoenolactone. And by adding this inhibitor, okay, to the transgenic mouse, we could completely ablate the arrhythmias. Not only can we completely ablate the PVCs, we could also completely ablate ventricular tachycardia. So these were interesting experiments, and I think to us showed the importance of IPLA2 beta, the potential importance of IPLA2 beta in cardiac electrophysiology. What about the structural biology of IPLA2 beta? So, as you can tell, IPLA2 beta is my favorite son.
Do you know anything about the mechanism of these VTs or PVCs?
I'll get to that with a couple of slides that Igor did, that little bit downstream, and then you could help me deconvolute what they mean. So, what's the first step in structural biology? Right, it's crystallizing your protein. So I'm not an X-ray crystallographer, but we collaborated with Sergei Parvalov, and we made a lot of protein and we put it in the refrigerator overnight with a number of different ingredients in it. And these are the first crystals of IPLA2 beta. It worked the first time, and it made beautiful crystals. We were incredulous. After 5 years of beating our head against the wall, we're able to crystallize a protein, overnight, in these needles that, we were sure would go on to yield the structure. But alas, I found out from one of my colleagues that the prettier the crystal, the more likely the diffusion pattern was poor. So, right, having pretty crystals is not necessarily a good sign. We went on to make hundreds, if not thousands, of more crystals, none of which diffracted properly. By properly, I mean, with better than 5 or 6 angstroms resolution. So we did the next best thing, was immigrate to NASA. And we thought that if we tried to crystallize this, right, in 0G, we'd be able to get better diffraction. So we convinced NASA to put IPLA2 beta on board in a crystallization setup, right, that we gave them. Okay, but unfortunately this didn't work either. So, this is pretty extreme, right? What are you going to do trying to crystallize a protein, right? So, and then one day, an undergraduate named Konstantin Malloy walked into Sergey's lab and said that he thought this protein was pretty interesting and he'd like to try and crystallize it. And like within a week, he had a crystal and a structure. He had no rhyme or reason why, like it worked in his hands. But they put seleniums on the thionines as we had done, and like some people just have golden hands, I guess, right? So it's the Midas touch for crystallization. So we could learn a lot from the crystal structure. Okay, these are the anchoring domains, which I feel very strongly interact with ion channels or accessory proteins to ion channels. Over here is the ATP binding domain, shown in the linear structure here. Okay, and then this is the catalytic side. This protein exists as a dimer, as we predicted from our gel filtration studies in the 80s. Okay, and you can see two views of this dimer. One looking down, okay, and one looking across. Okay, so here you can see the dimer and the catalytic faces ready for cooperative interactions, and the anchoring domains ready to interact with their accessory proteins. So at this point it became very clear that we needed to know how to measure lipids properly, just like it's important to measure ion selectivity and currents, okay, and lipids and lipid chemistry. There was no way to do that with complex zwitterionic lipids. And those of you who had tried in the past with old TLC plates or freshly purchased TLC plates know the difficulties in getting accurate quantitation. And we were blessed by the fact that we came up with a method that basically could analyze the phospholipid composition of major constituents within about 2 to 3 minutes, instead of having, like this last for days to weeks and having like 100 to 200% errors. Those are considered pretty good reproducibility, right? If it's like within an order of magnitude, it's not so far off. But Scheimpfen lipidomics consists of 4 parts. First, we realized that if we multiplex the extractions and the chemistry, we could get rid of chromatography in entirety. So you could just slop some organic solvent on top of your extract, and it'd extract all your lipids. Then you could directly infuse this extract, and by placing a different charge on the ion source, you can use that as a de facto column chromatogram and separate things as they came out of the mass spectrometer. And then bioinformatics, of course, to deconvolute, right, the different peaks that you saw using the different multiplex conditions. And this process was facilitated by the fact that you had mentioned that you had done some work in proteomics. So proteomics in some ways is much more difficult than lipidomics, and lipidomics in some ways is more difficult than proteomics. You ask a person in one field, they'll say the other field is much harder. But to a chemist who can recognize the fact that these have easily understood fragmentation patterns, right? This was a piece of cake. This was a walk in the park, right? You knew how these molecules were going to fragment, right, before we even saw the experimental results. You knew that SN1 would fragment, SN2 would fragment at the OX bond, OX2 bond, the polar head group would come off, or sphingosine-based phospholipids, you would have neutral loss of phospholipid. So this knowledge about the fragmentation patterns was put to use in a bioinformatics format, and greatly facilitated our ability to identify different properties of the enzyme using either a precursor ion scan or a neutral loss scan. And this whole analysis, if you just dump a bunch of chloroform in, centrifuge it, and then put it into a triple quad mass spectrometer, it takes about 10 or 15 minutes, if you're focused on that one sample. There's now software that can do this, but unless the products are manually curated, I'd be very suspicious, right, about any untoward or unbelievable claims. The software still has a long way to go in development. As many of you know, I've been studying diabetic cardiomyopathy, and the reason we got into diabetic cardiomyopathy was that diabetic cardiomyopathy has a fundamental difference in substrate utilization compared to non-diabetic heart disease. In diabetic heart disease, okay, about 95% of your substrate is fatty acid. So what's the difference between using fatty acid and glucose for substrate? So this has been probably the talk of hundreds of lectures in the last year alone, right, at WashU, more or less. So fatty acids generate more energy, but they also require more oxygen, right? Glucose generates less energy, but it carries its own oxygen atoms with it. So if you're in an ischemic condition, right, fatty acids are not so cool. Glucose can be used much more efficiently. And now we're going to see if this technology, the shock and lipidodynamics technology, could give us any insight into diabetic cardiomyopathy. So let's see what we found out. So this is a control heart, and we're looking now at acylcarnitines. Acylcarnitines are an intermediate in fatty acid metabolism, catalyzed by the production, enzymatic production by CPT1, carnitine palmitoyl transferase 1. Okay, and you can see that this is standard over here, this big P. And happy mitochondria, functioning mitochondria, have very low amounts. And some of this may be just, like an artifact of taking the heart out and homogenizing it. I mean, we can't do it instantly. But it's very clear that there's about an 8-fold increase in acylcarnitines in diabetic mitochondria. And then when we cause diabetes and then treat with insulin, it goes back to baseline. So having acylcarnitines in your coronary effluent, okay, or in your serum, will show, are a good marker of mitochondrial dysfunction. And mitochondrial dysfunction can be manifest through multiple mechanisms. So let's take a look here at now, this is a one-dimensional plot. Let's take a look here at multi-dimensional mass spectrometry. Okay, and what we have here is the control mouse on the left and the diabetic mouse on the right. This is the molecular ion tracing, and this is the triglyceride area right over here. Let's compare it to the diabetic triglyceride area. What do we see? We see about a 5-fold increase in triglycerides, right, in the diabetic hearts compared to the control hearts. Okay, so this was, pretty good confirmation that there was, there were too much lipids that were present in the heart, and generally the reason why lipids accumulate in the heart is due to mitochondrial dysfunction. Right, the mitochondria are not working well enough to chew up all the fatty acids that are transported, and instead they end up in a reserve storage pool, triglycerides, which are fine if you're an athlete and you have triglycerides in your heart, but not so good if you're a diabetic and have triglycerides in your heart, for reasons which we could discuss later. The other remarkable thing about two-dimensional spectrometry is you can see here, I want you to compare the arachidonic acid, right, in triglycerides in the normal heart compared to the diabetic heart. Whole new molecular species. In fact, the triglycerides are a historical record of what the hearts have been exposed to in terms of substrate and metabolism. So by looking at TAG content, triglyceride content in molecular species by multidimensional mass spectra, you can see that there's a huge difference in arachidonate, and arachidonate is mobilized in diabetic cardiomyopathy and ends up in the triglyceride pool because it has nowhere else to go. Well, there's another molecule that we worked on, and this was also through first developing the technology and subsequently looking at the physiological effects. So cardiolipin is basically a dimer of phospholipids. So here's a phosphatidylgroup that's connected by a glycerol molecule. Cardiolipin is only present in hearts, is only present in the mitochondrial membrane. All mitochondrial membranes contain between 5 and 15% cardiolipin. If you're a heart where 30% of volume, right, is mitochondria, this can represent a substantial quantity of total phospholipids. Cardiolipin is known to be essential for complex 4 function in the electron transport chain. It adopts an inverted hexagonal phase, promoting mitochondrial fusion, efficient in mitochondrial dynamics. It binds to cytochrome C, and if you don't have cardiolipin there, the cytochrome C will be released, which can trigger apoptosis. And it facilitates the transduction of a proton gradient into an electrical potential. So we came up with what I think was a rather clever idea, is cardiolipin is really the only doubly negatively charged phospholipid that is present in substantial quantities. So when you look at the mass to charge value, right, you'll be looking at 1 charge per molecular weight, whereas cardiolipin now has 2 charges per molecular weight. So since each phospholipid contains C13 in it, right, 0.078%. I'm sorry, not 0.78%. Then you could basically look for doubly negatively charged molecular species, and those almost by definition have to be cardiolipin. And what happens to cardiolipin in diabetes? Well, there are huge changes. So this is a control heart, and you see how we now have the peak start? So this is the n+1/2 peaks, right? Indicative of the presence, right, of a doubly negatively charged species. Okay, this is a control heart, and this is streptozotocin-induced diabetes. And this is even more magnified, okay, in OB mice, okay, where you see about an 80% loss of cardiolipin in the mitochondria, which is accompanied by vast changes in mitochondrial function.
I'm sorry, what's the next circle?
Oh, so this is also cardiolipin, and these are now polyunsaturated fatty acids that are now in cardiolipin. Usually cardiolipin has only a little of 20:4, 22:6, okay? This is mostly 18:2 fatty acids. Okay, I've realized I've broad brushed a lot of this, but I'll be happy to entertain questions and details at the end. So here we have 18:2 cardiolipins, and then here we have cardiolipins that now have polyunsaturated, like 20:4 arachidonate or 22:6 docosahexaenoate. Okay, so here is an example of now CPLA2 gamma that was generated by overexpression using a cardiac myocyte-specific promoter, the MHC promoter. And now this is work that was done by Igor and colleagues, and maybe some of you in the room. The overexpression of CPLA2 gamma dramatically changed the longitudinal and transverse conduction velocities and the anisotropy of conduction. This is in blebbistatin-treated hearts and with methods that you guys know much better than me. And this is looking for the location of the PVCs in these phospholipase overexpressing hearts. And you can see that most of the PVCs come from the apex. Okay, the incidence of arrhythmias in this model. Okay, so these hearts are damaged from the beginning, right? So these hearts have lots of VT and VF, even in the non-ischemic state. Okay, and then the real difference, right, occurs basically in reperfusion. Okay, where you can see this like enormous difference in the CPLA2 gamma transgenics, which really doesn't decrease much, although it is lower than control, and reperfusion work basically goes away. These are examples of fast pacing induced VT-VF with PVC. And this is mapping, okay, that you also probably have seen many examples of. This is with an overexpressed phospholipase, and you can see that the VT is contiguous over the 5 examined zones. Okay, moving on. I'd like to tell you about some recent work that we've done. I mean, we started this with a slide from 1983, and now we're going to go to 2016. So it used to be thought, and I told you that, you know, arachidonic acid was specifically oxidized by a bunch of enzymes that are present in the heart. And using molecular modeling, we basically examined cyclooxygenase 2, and we docked cyclooxygenase 2 with arachidonic acid. And amazingly enough, lysophosphatidylcholines containing arachidonic acid docked even better than arachidonic acid itself. Okay, so what this led to the obvious experiment, right, going from in silico, right, to an experimental result. Well, why don't we add 2-AA-LPC, 2-arachidonyl LPC, and see if anything happens with COX-2. This is LPC docked here. This is the tyrosine 386, which abstracts the C13 hydrogen as a radical. Here's 386. And there's actually tighter binding in the polar head groups than with arachidonic acid alone. Very amusing. So this slide says a lot and shows you that it's dangerous to take a bite out of that apple because it's not only arachidonic acid, okay, that is the substrate for cyclooxygenase 2, as people have thought, and there are 38,000 papers on cyclooxygenase 2 in PubMed. Not one thinking that there was another substrate that was active. This is control. This is COX-1, which has a little oxidation associated with it. And this is cyclooxygenase 2. What we're making here, and let me orient you, okay, is this is the phosphocholine head group. Okay, this is the SN2 arachidonic acid that now you see is now hydroxylated. So now we have hydroxylysophospholipid. Okay, and you could hydroxylate it, or you could add, you could cyclize it for prostaglandin, or you can make an 11-heat LPC, which used to be thought of as adventitious oxidation product, I believe is the language that was called, but actually it's stereo-specifically made, as you can see a couple of slides down the street. And this now is with LPE. LPE doesn't make 11 heat, as far as we, right, I mean, maybe, but not very much. What it mostly does is it cyclizes the arachidonic acid into a prostaglandin. Again, control, cyclooxygenase 1, and cyclooxygenase 2. So the profiles of these different lines of lipids as oxidation products vary, right, as the structure of the head group varies. And now looking for stereospecificity. If this was just a question of cyclooxygenase making, free radicals, then you wouldn't expect any R versus S selectivity, right? The equal amounts. And here we've separated R versus S using a chiral column, and you can see there's only really one isomer formed in at least 90% moment of excess, okay? This is the commercially available racemic mixture, commercially available chiral compound, and the experimental results. Okay, so it only makes R, 11 heat. It only makes it makes a mixture of about 70-30 R versus S, 15 heat. Whether this is actually an enzymatic product or just, from free radicals traversing, carrying on or having a prolonged lifetime, I can't answer with certainty, but clearly there's a 3 or 4 to 1 predominance. And here we look at PGE2s. Now, these have 2 chiral centers in them, so they're by definition diastereomers. And these diastereomers make only one form of COX-2. So this is all the possible PGE2s that are made. This is authentic PGE2 in the naturally occurring chiral state. And this is what the enzyme makes, right? Stereospecific PGE2. Now, let's take this one step further, okay, into now looking at what happens in the cell type. This is fibroblasts from a mouse lung. Using the IPLG gamma knockout mouse that we generated. So you can see here that stimulation with ionophore causes about a 2-fold increase, which is substantially blocked, right, in the mouse that has the IPLG gamma knockout. This is yet another phospholipase, right, that we have been working with both in transgenic and knockout forms. And then we can see over here, so this, look at the y-axis, this is picomoles, okay, and this is picograms, okay? So you make about, in a stimulated cell, you make about 600 picomoles per milligram of protein, but the amounts here, right, is picograms, so it's about 1/500th the amount, Okay, so only one molecule of 500 or so that are released actually goes on to oxidation, which is very highly blocked by knocking out IPLAG gamma, both in terms of generating 15 heat LPC or 11 heat LPC. Now, a lot of people are very upset with me because not only does this work with choline phospholipids, it works with inositol lipids, it works with ethanolamine lysophospholipids, it works with phosphatidic acid. So we've just expanded the complexity of metabolomics by probably a factor of 50 to 100. So I don't have so many friends anymore because and I think we've also put to bed the notion, right, that arachidonic acid is the only substrate. In fact, now we're seeing very good oxidation with lipoxygenases on parent phosphatidylcholines and lysophosphatidylcholines presented in bilayer form. And this also has more far-reaching observations, again, allowing me to make more enemies, showing that the products that they've been studying can be made by a previously unknown pathway. And here we have two AALPC that can be operated on by each of these three pathways, okay, and then it can be cleaved to make either PG glycerols, okay, or E glycerols, which are the cannabinoids, which, right, if it's arachidonic acid, or the cannabinoid ligands for the cannabinoid receptor, right, who's very disturbed about this field finding, or the growth factor, right, field with LPAs, with lysophosphatidic acid. So instead of cleaning up a problem, I think we made more of a mess in a certain way. Life used to be simple when the apple was passed, but now it's become quite complicated, and there are many more years of work to be done. And this is sort of the cartoon, I guess, simplifying everything. With COX-2, right, this is the crystal structure from the protein data bank, with the lysos going in and all these novel phospholipids going in. And there are over 30 head groups that you can have over here, and to date we've not found one that can't do this oxidative process. So life is much more complicated than we had previously thought. Okay, so it's been a long-standing goal, right, to decrease infarct size. And because IPLA2 gamma is a mitochondrial enzyme, and because in previous work we showed that IPLA2 gamma was necessary for opening of the mitochondrial permeability pore, we wanted to look to see if knocking out IPLA2 gamma had a salutary effect. So we generated a cardiac myocyte-specific IPLA2 gamma knockout. You can see here that in the northern there's no RNA, and you can see the selectivity here, heart wild-type knockout. These residual bands, the heart, of course, has fibroblasts and endothelial cells, right? So it's not surprising you'll see some shadows. This probably is a false positive band. And the other interesting thing to note is the different patterns, right, of IPLA2 beta in heart, in skeletal muscle, kidney, and brain, right? So there's tons of more work to be done with that. So we take our knockout, and we show that our knockout basically decreases activity by about 80%. Now, we don't know if the other 20% is coming from a compensatory effect or exists in its natural state. That's one of the reasons why you have to be very careful with knockout animals, right? You don't know whether what you're looking at is a compensation to dramatic genetic alteration, right, or is part of the naturally occurring molecules and activities in cells. So IPLA2 gamma knockout decreases the MPT opening probability, and here you can see this is with calcium, okay, I'm sorry, without calcium, and the blue is with calcium. This is calcium plus cyclosporin, okay, and this is with palmitic acid, calcium plus palmitic acid. You can see palmitate in the plane of the membrane is known. Leninger described this, palmitate opening the mitochondrial permeability pore in like the 1960s. And this palmitate, whoever's measuring palmitate here, needs to be very careful because palmitate's everywhere. We measure palmitate now as the derivatized products, and you can't measure it because it's like You know, it's on the desk, it's floating around in the air. Mass spectrometry is so sensitive that you need to be very careful when you make when you measure palmitates. And a lot of what people have called palmitate toxicity probably is investigator misguidance. But you know, like that is or investigator confusion, however you want to put it. The other interesting thing about the heart-specific knockout, okay, so this is wild type, no ischemia, knockout, no ischemia, red and blue, okay, and then compare it with the amount of heats, right, that are made, okay, in the wild type after an ischemic episode. And this is actually, this was done by Carlo, so this is in vivo myocardial ischemia. I mean, the real thing. It's not like hanging a heart on a cannula, not to, you know, or putting in a blebbistatin. This is an in vivo heart that Carlo left ischemic for 30 minutes and then opened up the flow. And then Dana Abenshine measured the area, the left ventricular volume, the area at risk, and the infarct area. And the numbers are shown here on the right. So there's about the same area at risk per LV. The infarct area was greatly reduced, about 60 to 70%, and the area at risk compared to the area, the infarct area compared to the LV, as well as the infarct area compared to the area at risk, was markedly decreased. And that's shown in pictorial form over here. And finally, we're going to go and make the transition that Bureau has done with his vest and look at human hearts, which I assure you have very little similarities in many ways to human hearts. And people have basically discarded the importance of mitochondria in human heart failure because several very prominent investigators have measured using high-resolution respirometry both control and fouling heart samples from ventricular assist device placement. And as you can see, the oxygen consumption in the presence of state 3 with ADP, succinate, rhodamine, or TMPA, and ascorbate, which is an electron generator, okay, it's basically the same, right, in heart failure, right, versus controls. Or as people make you say now, in non-failing hearts compared to failing hearts. And now we're going to look, okay, again at the mitochondrial swelling, which is an indicator of MPT opening. Okay, and you can see a very large difference, okay, in the control hearts compared to the failing hearts. Okay, so there is differences between these mitochondrial preps that are not visible by high-resolution respirometry. And for people to tell you that mitochondria are not involved in energy transduction or the symptoms and stigmata of heart failure, they're completely wrong. They are very easily observable. All you have to do is put the mitochondria in a UV spectrometer and measure the swelling. Right? What could be simpler? No mass spectrometry needed. So you go ahead and you look at these, and you can see the mitochondrial swelling is up. It's not due to a difference in cyclophilin or the nucleotide transporter or VDAC. They're all the same. But this is what we found to be really interesting, right? And the control hearts, okay, here we give we incubate them either in EGTA or in calcium, okay, or the failing hearts in EGTA or calcium. So as you can see, there are responses in the controls. But 1415E, which has previously been implicated in myocardial damage, okay, by Falk and colleagues, as well as a number of other investigators, you can see that the failing heart doesn't do very well in making cytoprotective beats. Moreover, you can see that the failing heart just makes a ton of 15 heats. And as you'll see in the next slide, these heats, okay, are basically diminished in the presence of bacoline, which is an inhibitor of the lipoxygenase pathway. but not the epoxygenase pathway. Okay, and you can see the difference here. So this is red, okay, with calcium, blue with cyclosporin right over it, with EATs, okay. You can see that it basically rescues it. Okay, and then we're going to go down to bacillin. Okay, and bacillin now is working on the lipoxygenases. So here we have 5 heat, 12 heat, 15 heat. Okay, and say with bacillin, you have very little swelling in the presence of calcium, implicating the lipoxygenase. Now here's what's really interesting. So if you had to like say to me, you know, like if you asked me, you know, like what would I expect in heart failure? Would I expect the heart to have more arachidonic acid and make more arachidonic acid due to inflammatory cell inflammation? I'd say, of course, it's going to be more arachidonic acid made. But no, it's exactly the opposite. In healthy or non-failing hearts, in control hearts, they make a ton of calcium-stimulated arachidonic acid. Okay? In failing hearts, they hardly make any. Same thing is true with LPC, right? In healthy hearts, or non-failing hearts, you make quite a good amount of calcium-stimulated LPC, the co-product of the reaction. But in failing hearts, hardly any change in it. So arachidonic acid has bidirectional effects. It both helps the heart if it's in the right place, or hurts the heart, okay, if it's in the wrong compartment. And you can see that in control hearts, you get this rapid rate. This is an initial rate enzyme reaction, okay, and easily a 5-fold difference, okay, in the amount of arachidonate production, presumably going to the generation of making healthy icosanoids. And this is even clearer in this slide, which shows mass spectra. Here we are at time 0. And these are just derivatized fatty acids, 16O, this is dideutero, internal standard that we use over here. And this is 10 minutes in EGTA, okay, and you can see that these guys didn't change so much. And you add calcium to control heart, and you get this huge increase. Nothing happens in the failing heart. I mean, this was like data that was very surprising, if not shocking. Okay, to the people, both to myself and the people who generated it. So, what is this mysterious protein? So, you know, like I'm sort of an old school biochemist, and you know, I believe in the axiom by Arthur Korberg: first purify, then think. Right? Unfortunately, purification is very painful. But we did our first column, and we found conditions that could, you know, preserve human heart activity, and students were all excited because look, it comes out in a single peak, right? And then you take the single peak and put it on a cation exchange column, and it comes out as 20 other peaks. And they go, oh, but now we have it here, right? It's this single peak over here. Well, we take that single peak and put it on a mono Q column, and they go, oh, it's this thing over here, you know, from the 20 peaks. So now we're 30,000-fold purified from human myocardial homogenate. Okay, and we look at the mono Q fractions, okay, and this dash line over here basically is shown after labeling with activity-based profiling and antibodies to biotin. And this sequence, now by proteomics, turns out to be CPLA2 zeta. So this is the first role of CPLA2 zeta in any system that I'm aware of, and the human heart is full of it. Where that leads us remains to be determined. But I'll leave you with a philosophical slide. So when I went to boarding school, the alumni were very interested in winning football games and championships. So each year they brought in what we called ringers, you know, people from, you know, like different schools, high schools, that wanted to go to an Ivy League college. So you didn't know people on your football team, right? They were just brought there for a year to play football. And Mount Hermon, which was the school I was from, and Deerfield were both 7-0 going into the eighth and last game of the season. Okay, and this is exactly what's happening today, 50 years later. Okay, so people are playing football while the science building burns. right? I mean, if I had only known. And I'd like to thank all my collaborators and students, and thank you very much for your attention.
Thank you for, this is really a tour de force, and many techniques and a lot of data, and you covered a lot of grounds. And I was interested in the mechanism, the arrhythmias that result as a consequence of all of this, but that's not clear to me. Maybe you can put this slide of the arrhythmia on just
with the optical stuff, or
the optical stuff. I'm curious to see if I can. Yeah, the one before.
Yeah.
But these are not arrhythmias. These are paced, just to measure velocity.
Right.
And
these are.
No, go back. Let me see. So the velocity.
Yeah, the velocity is slower, right, in the genetically manipulated animals, right, and the anisotropy is larger in the genetically manipulated animals.
Okay, and then the arrhythmias.
The next slide?
The next one.
The next one?
These arrhythmias. No. So I thought these arrhythmias were calcium-based. So if they're calcium-based, they're probably focal in mechanism. It's hard to tell from these maps. Shows some reentient activity, but it's not clear. Hard to tell. It could be either way. Okay, I'm sorry.
I noticed that the last set of slides were all, the dates of the publication was like 2 weeks ago.
Yeah.
No, I don't, and so I was curious, what did the reviewers say?
So we sent it first to JCI, and the reviewers at JCI said, well, you know that that IPLA2 gamma prevents opening of the MPTP. So why are you surprised when it decreases arrhythmia, when it decreases infarct size?
That's not a good question.
That was a classic. And then for the crystal structure, that was like that was canned at Nature, and the comment was it's clear that the crystal structure is not the physiologically active form of the enzyme, because the IQ complex is distal to the other calmodulin binding site, which no structural biologist would ever say, because obviously proteins are dynamic entities, and binding to membranes cause allosteric and conformational changes. So I don't want to make the students too disappointed. I mean, so we've gotten a lot of good reviews too, but people don't like things to be too far from, like where they are too. There's an impedance matching that is important. Yes?
So what is the mechanism in your gain-of-function model for the your alpha MHC gain-of-function model for the phospholipase? Do you think that you're just conditioning the animals to, conditioning their metabolism, or is it a direct interaction in the phospholipase with certain ion channels?
Right. So in the IPLA2 beta, right, for the arrhythmias there, you may recall that the wild type at time 0, right, was really quiet, and it was ischemia-induced arrhythmias. And there is ischemia-induced activation of IPLA2 beta. Okay, and that's probably due to the accumulation of acyl-CoA, right, which then reverses the inhibition by calmodulin. That's a 4-step. Are you okay with that?
Yeah.
Okay. But it's like a competition between, inhibition and activation. And the other thing that I didn't talk about, but we have a lot of data on now, that many of the sites around the calmodulin binding domain are phosphorylated by CAM kinase, which also can bind to one of the arms of anchor. So the story just keeps on getting, you know, it's not, I don't feel like I've solved any problems. I feel like I've created layers of difficulty.
We all do that.
Well, I was looking for a straightforward path, right? And instead I found more doors.
Did anybody ever look at the role of antiarrhythmics in this hospital? When you treat arrhythmias with antiarrhythmics, do they change the phospholipid profile? Or what do
I don't know. So when they would have looked at this was before, right? This would have to be recent work within the last 15 years. And I think people have lost interest in, you know, like lipids in the last 15 years in terms of arrhythmias. Because all the measurements of lipids before, right, that preceded the 15-year and the shotgun lipidomic stuff were, right, who knows what they were measuring.
So do you think that's a useful question to ask?
Sure. Why wouldn't it be useful? I'd love to know, but I mean, I try my best to, you know, like motivate and animate and kinetically stimulate, you know, my students, and they've done a lot, I think, but you always want more. Yes?
So can we go back a little bit to the PLA, the calcium CAM and acyl-CoA, kind of that competition there? So you kind of also looked at ATP. Can you elaborate a little more on how ATP and CAM is regulating this?
Okay.
The mechanism by which CAM and ATP is regulating the protein structurally?
This is a slide
Right.
that you want.
Yes.
Okay. So here we have calcium activated calmodulin. Okay, so calcium causes calmodulin to form like this dumbbell.
Right.
Right? And the activated calmodulin, okay, can bind, okay, to the IPLA2, right near the IPLA2 active site. Okay, but in the presence of acyl-CoA, okay, acyl-CoA displaces the calmodulin. Okay, it's not only acyl-CoA, it's non-hydrolyzable acyl-CoA. And we have a mass spec showing that cysteine 651 on IPLA2 beta interacts with the serine at the active site.
Okay.
Does that help at all?
Yes.
Maybe?
Okay, any other?
Thank you very much.